Let's study about the viral replication. The purpose of viral replication is to allow production and survival of its kind. There are 8 stages of viral replication: adsorption, penetration, encoding, expression, replication, assembly, maturation, release. To initiate infection cycle, a virus must first recognize and bind to a suitable host cell. Virus attachment consists of specific binding of a virus attachment protein to a cellular receptor molecule. Receptors are specific cell surface component of the host to which the virion attaches. No plant virus is known to use a specific cellular receptor of the type that animal and bacterial viruses use to attach to cells. Penetration of the target cell occurs in a very short time after attachment of the virus to its receptor in the cell membrane. Cell penetration is an energy-dependent process, that is, the cell must be metabolically active for this to occur. There are 3 different mechanisms of penetration. Translocation of the entire virus particle across the cytoplasmic membrane of the cell. This process is rare among viruses. It must be mediated by proteins in the virus capsid and specific membrane receptors. Endocytosis of the virus into intracellular vacuoles. This is the most common mechanism of virus entry into cells. It does not require any specific virus proteins. It relies on the normal formation and internalization of coated pits at the cell membrane. Fusion of the virus envelope with the cell membrane. It requires the presence of a specific fusion protein in the virus envelope. For example, influenza hemagglutinin. These proteins promote the joining of the cellular and virus membrane, which results in the nucleocapsid being deposited directly in the cytoplasm. Uncoding, the events that occur after penetration, in which the virus capsid is completely or partially removed, and the virus genome exposed in the form of a nucleoprotein complex, which makes viral nucleic acid available for transcription to permit multiplication to proceed. In this stage, the infected cell starts showing symptoms.
Viruses must first get into the cell before viral replication can occur. The purpose of viral replication is to allow production and survival of its kind by generating abundant copies of its genome and packaging these copies into viruses. The virus is able to continue infecting new hosts. The process of assembly involves the collection of all the components necessary for the formation of the mature virion at a particular site in the cell. During assembly, the basic structure of the virus particle is formed. The site of assembly depends on the site of replication within the cell and on the mechanism by which the virus is released from the cell, and varies for different viruses. Maturation is a stage of the life cycle at which the virus becomes infectious. Maturation involves structural changes in the virus particle that may result from specific cleavages of capsid proteins to form the mature products during assembly. Virus-encoded proteases are involved in maturation, although cellular enzymes are used in some cases. Virus is released where it can infect new cells and repeat the process. This is the basis of virus spread, whether from cell to cell or from individual to individual. Here is shown the complete process of viral replication.
Do subscribe the channel for more videos.